So hello students, welcome back to Vedantu NCERT Solutions. And in this particular channel, we do NCERT line by line. We do NCERT solution to make sure your transition into studying the NCERT becomes very easy for all of you, right? So with that being said, the chapter for today, right? We have one second, please. We have chapter for today is going to be biomolecules. It may seem like a difficult chapter, but let me tell you, it is a very easy chapter. So let's try to understand the lines of NCERT. Anybody you feel any doubt, you can always ask me and I will reply to it. So let's start the class. Biomolecules. What is the meaning of a biomolecule first of all? What is the meaning of a biomolecule? Bio means living, right? Bio simply means living. Yes. Biomolecules. Bio means living, right? Bio means living. Molecules are the molecules. So living molecules are called as biomolecules. Now these biomolecules can be your proteins, carbohydrates, lipids, and amino acids. All of these or the following are basically the biomolecules, right? By different types of biomolecules. There is a wide diversity in living organisms in our biosphere, right? This is the Earth, students. If this is the Earth right here, on this earth we have wide variety of organisms. For example, you could have millions, more than 3 lakh plant species of flowering plants only. More than 3 lakh flowering plants there can be. Or at the same time there can be so many different of animals, insecta, the phylum. So there are so many different types of diversity basically on the earth. Now a question that arises in our mind is that all living organisms made up of same chemical? Simple question. Now tell me, do you think the plants and the animals or the microbes, is every single one of them made up of same type of molecule, or is there a different type of molecule for each plant? So if I tell I am made up of carbon, you are made up of silicon. Can I tell that? No, I cannot tell that. So every single organism is made up of a basic framework, same framework. It is elements or compounds. You have learned in chemistry how element analysis is performed. If we perform such an analysis on plant tissues, animal tissues, and microbial paste, we obtain elements like carbon, hydrogen, oxygen, and several other and their respective content per unit mass. So basically what I can tell you here is in the next line here is it could be microbe, it could be a plant, or it could be animal. Every single organism is made up of few elements. A few elements such as carbon, hydrogen, and oxygen. Organic compounds every single organism is made up of. It could be plant, it could be animal, it could be microbe. Okay. Now, the next thing you need to understand here is if this is the Earth, the outer layer of Earth, students, the outer layer of Earth is called as crust. Now, students, if you take a piece of Earth crust, if you take a piece of Earth crust, and if you do the chemical analysis, if you take the Earth's crust and if you do the chemical analysis, you will obtain something called as carbon, hydrogen, oxygen. Now, didn't we just learn the carbon, hydrogen, and oxygen? They are present in the case of your plants as well as animals and microbes. Now I'm telling they're also present in Earth's crust. Now you might ask me, sir, then what is the difference between living and non-living then? Living things also have carbon, hydrogen, and oxygen. Non-living things also have carbon, hydrogen, oxygen. Then what is the difference between the plant and and Earth's crust then? Students, the answer is very simple. The answer lies in the abundance or the quantity. For example, the same oxygen which is present here and the same oxygen which is present here, in the case of living things, oxygen will be in a higher quantity. Oxygen will be present in a very high quantity here. Amount, concentration will be more. At the same time, if you look at the carbon also, carbon in the case of your living cells would be higher. Same goes for hydrogen. So in the case of your living cells, the amount or the concentration of your hydrogen, oxygen and carbon is comparatively higher when we compare to Earth's crust. Earth's crust is comparatively lower. Okay, lower. Abundance is lower. See, it's given you NCERT also. So if the same analysis is performed on a piece of Earth's crust, an example of non-living matter, we obtain a similar list. Then what are the difference between the two lists? That is, what is the difference between living and non-living? Right? In absolute terms, there is no such difference. There is no such difference between them. Same carbon will be present in both. Same oxygen will be present in both living and non-living. Then what is the difference then? All the elements present in the sample of Earth's crust are also present in the sample of living tissue. However, a closer examination reveals that the relative abundance, see, relative abundance, and where is the abundance higher? Abundance is higher in the case of living. Of carbon, hydrogen with respect to other elements is higher in the case of living organisms. So, very simple, students. In the case of living tissues, like such as your plant cell, animals, as well as microbial waste, every single one of them is made up of carbon, hydrogen, and oxygen. The same goes for Earth's crust. The Earth crust is also made up of carbon, hydrogen, oxygen. Okay. Then what is the difference? The difference is the abundance. In the case of living, we will have more. That's all. Living will have more abundance. Here we'll have very less or lower abundance. Understood? This is what is given here also. Can you see? Comparative analysis. Hydrogen is 0.14 in the case of Earth's crust, but 0.5 in the case of human body. 0.03 of carbon, 18.1 carbon in the case of human body. More here, always more, more. At the same time, if you look at the oxygen also, in the case of your human body, oxygen is more when you compare to the Earth's crust. That is the major difference between living and non-living things. Even though they're made up of same material, there is a difference in the abundance. Difference in the abundance. Now, students, I'll teach you one small experiment. Let's see how many of you understand the small experiment now. Okay. Experiment is very simple. Experiment is very, very simple. What you need to do is understand. We will take something called as one leaf. We will take one leaf here. We can take a leaf, or we can take also take a liver piece also. Let's take one rat liver piece here. Rat liver piece. Liver piece, or this is a leaf. You're supposed to take any one of them, any one, not both, any one, either leaf or liver. Don't take both and all. Too much of work will become. You need to take any one of them and add it into a mortar and pestle. Add it into mortar and pestle. Then once you add it here to this mortar and pestle, you need to add some form of solvent. Solvent. The solvent which I'm talking about here is nothing but trichloroacetic acid. Trichloroacetic acid. Now, what to do next? Next step, make a chutney. Take the trichloroacetic acid, take the leaf, and nicely make a chutney out of it. And we can call this chutney as slurry. This is trichloroacetic acid, okay? Doesn't even look right. 1 second, friends. Now, once you make the slurry, the chutney, this is chutney, okay, slurry. You need to take the slurry and add it into, take the slurry and add it into a separate type of beaker. For example, here I have a beaker now. Here I have a beaker. I have a beaker here. To this I am adding this. To this beaker I am adding it, okay. But I cannot add it as such. You need to filter it. So on top of this I am keeping a muslin or cheesecloth. Muslin or cheesecloth. You might have seen, no, when your mother is making chapati, you take the atta and you sieve the atta, right? Sieve it, right? Sieve it. So here we have muslin cloth. Now we are adding a chutney on top. Let's add the chutney on top. Adding a chutney here or a slurry. Now, after some time, since the muslin cloth has small, small holes, some amount of chutney will go down. Some amount of slurry will go down and settle on the bottom. But at the same time, some slurry will be left on the top, which is too big to go through the muslin cloth. Okay. Now, interesting thing note here. This part, which is present on the bottom, it was able to come down because it was soluble in the trichloroacetic acid. That is why it is dissolved in the acid, is able to come down. So I will call it acid soluble fraction. Acid soluble fraction. Also called as filtrate. Also called as filtrate. YouTube sometimes plays games with all of you. Okay. I can also call it filtrate. I can also call it filtrate. Now, what about the size? The all the chutney which is coming down, okay? All the chutney which is coming down, the size is obviously lesser because it is able to get solubilize and it is coming down. So, can I tell the size is here less than 1000 daltons? The size is a little less than 1000 daltons here, okay? And I'm calling it micromolecules. Micromolecules. Now, what about which is present on the top? What about this? This part is called as acid insoluble because it is not soluble in acid. It is going to remain on the top. So I'm going to call it acid insoluble fraction. Acid insoluble fraction. I can also call it retentate. Call it retentate because it is retaining on the top. The size here is more than 1000 daltons. More than 1000 daltons. And I can call this macromolecules. I can call this macromolecules. So what we learned, students, we learned here is when you try to make the chutney, you get retentate and filtrate. Retentate on the top, more than 1000 atoms, 1000 called macromolecules, acid insoluble fraction on the top. The one which is coming down, the one which is coming on top, bottom is called as acid soluble fraction or filtrate, or also called as size less than 1000. We can call it micromolecules. Okay, I will check. I will check Telegram later on. Okay. Till you any doubt? Till you any doubt, students? This is a small experiment which is given in NCERT. Now, if you notice here, students, macromolecules examples, complex carbohydrates like your disaccharides, oligosaccharides, lipids, proteins, nucleic acids. In this, macromolecules, you have simple sugars like your glucose, amino acids, and nucleotide, small structures here. But students, here one imposter is there. Imposter. The imposter here is none other than the lipid. Lipid is the imposter. Why? Because lipid is very special because the size of the lipid is less than 1000 daltons. Even though the size of the lipid is less than 1000 daltons, it will be present where? Here it will be present on the top. We can find lipid here. We can find lipid here. We can find lipid here. So it is impossible here, even though the size of the lipid, even though the size of a lipid is very small, it's very cutie, it is lipid. Even though size is very less, very small, lipid will be present on the retentate. Why? Because the answer is very simple, because all the lipids are holding on to themselves. When all the lipids hold hands, they form one big gang. When they form a big gang, they cannot go down at all. They cannot go down at all, and hence the big gang of lipid is present on the retentate. It is present on the retentate. Okay. One second, students. All of you understood? Yes or no? So all the retentate, all the lipids are present on the retentate because they are forming bonds among themselves. That is called as vesicle formation. It's called as vesicle formation. Clear? Don't worry, notes will be given to you. I will send the notes today only because I'm home early today. So I will send all the notes today only. Okay, don't worry. Now, students, that is done. Okay, this is also done. Now understand what is given here. We can continue asking in the same way, what type of organic compounds are found in the living organisms? How does one go after finding the answer? To get an answer, one has to perform chemical analysis. We can take any living tissue, you can take one plant, leaf or liver, okay, and grind it in the trichloroacetic acid, CH3CO CCOH. using a modern pestle, we obtain a thick slurry, thick chutney we get. Okay. If we strain this through a cheesecloth, here we're doing here, see, straining it through a cheesecloth. This is cheesecloth. Cheesecloth. Okay, cheesecloth. Okay. One would be called as filtrate, which is on the bottom. More technically, acid soluble pool. So, filtrate, acid soluble pool. Micromolecules less than 1000 Dalton size. Okay. The second one is called as retentate on the top and acid insoluble fraction on the top. The scientists have found thousands of organic compounds in the acid soluble pool. Many organic compounds are found in the acid soluble pool here. Okay. Understood all of you? That was the first experiment. That was the first simple experiment to understand. Now, in higher classes, you will learn about the how analyze a chemical tissue, living tissue sample, identify particular organic compounds. It is suffice to say here that one extracts of the compound, then subject to the extracts to the various separation techniques, one has passed a compound from all our compounds. What I tell you here is, you have learned the basics now. Once you get the extract, the micromolecules and macromolecules, the filtrate and retentate, you take this filtrate and retentate and you add it into another different types of machines which will give you more and more information regarding the constituents or the what is present in the liquid. Okay, that's all. See, written here. Analytical techniques can be there. When applied to the compound, give us an idea about the molecular formula and the probable structure of the compound. Okay. Now understand the second experiment now, friends. Second experiment is called as the destructive method. It is called as the destructive method. Why? Because you're burning here. What are we burning? The entire plant leaf only. Leaf you're burning. For example, in this destructive method, you take one leaf. You take a fresh leaf. You take a fresh leaf. Okay. To this fresh leaf, you need to do what now? This fresh leaf will have water. Yes. So I cannot call it wet weight. It has water. Wet weight. Okay. That is water is present. Water is present. Okay. Now what do you do? You evaporate all the water. You do evaporation. That is minus H2O. Then what do you do? Once you do the evaporation, you are left with one more leaf. But this is called a dry leaf. A dry leaf. Now you take the dry leaf and completely grind it. After grinding it, you will get a powder. Take the powder and nicely burn it completely. Burn the powder of a leaf. Completely burn it. Now students, once you burn, what happens? You get ash. When you're burning it, it is undergoing oxidation. Oxidation. So my any carbon is there gone, any water is there completely gone. Okay, oxidation is happening. I am finally getting the ash. It's called as ash weight. Ash weight. Now you take the ash weight and you do analysis. You do analysis and you get the result of what are the inorganic compounds. See, understand students. If leaf, all the carbon compounds removed here only, all the water is been removed. Obviously, right now in the ash, you'll be only left with the minerals, different types of inorganic compounds now. Yes, that is what is happening. You're left with a lot of inorganic compounds. See here. Living organisms have also got inorganic elements. See, they also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive method has been done. That is, you take a when we have small amount of living tissue, say leaf or liver, and we call it wet weight starting. Then dry it. Then we got dry weight. Okay. The remaining material give us dry weight. Now, if possible, tissue is fully burnt. How? Nicely make a powder out of it and then burn it. Boom, boom. Okay. So when you're burning it, what is happening? Carbon compounds are oxidized to gaseous CO2 and water vapor are completely removed. So all the carbon compounds and as well as all the types of carbon and hydrogen is completely removed when you burn things. Okay. This ash contains inorganic elements like calcium, magnesium, etc. They are the different types of inorganic things present here. Inorganic things present here. Okay. Inorganic compounds like sulfate and phosphate, etc. are also seen in the acid soluble fraction. Okay. Therefore, elemental analysis gives us the elemental composition of living tissues in the hydrogen, oxygen, chlorine, carbon, etc. So, chemical analysis is basically telling you which type of organic compound is present, which type of inorganic compound is also present here in the case of your plants. Okay. So, inorganic elements, burning it. Organic elements, we have the normal test here. For organic compounds, we have this normal test. For inorganic compounds, we have the ash test, burning test. Okay. I hope the simple difference is clear to all of you. The simple difference between organic and inorganic compounds which you obtain is completely clear to all of you. Yes. Now, students, while analyzing the compounds, give an idea of for organic and inorganic constituents. It's very easy. Now, students, tell me if you're ready for amino acid now. Ready for amino acid? Yes or no? You're ready to learn amino acid structure. Ready? Friends, whenever you're studying, right, always keep some form of snacks next to you. So what I keep next to me is always and always I keep kaju, badam, and raisins. I always keep some form of nuts which will give me energy and which will always make sure that I can be proactive. Right. So always all of you also keep a small snack with you. So please don't keep biscuits and chocolates. Please don't keep biscuits, chocolates, or chips and all. Trans fat. Okay. No. Amino acid. It's always food is life, students. Food is amazing. Okay. Now, students, tell me what is CH4? What is CH4? Tell me. What is CH4? Anyone? What is CH4? All of you know basic chemistry and you can tell me what is CH4. CH4 is nothing but your methane. Is your methane. Now what you need to do is you see this methane right here CH4. You have to substitute one hydrogen with a different types of element to understand the structure of amino acid. For example, I will replace this hydrogen with a carboxylic group. I will replace this hydrogen with a amine group, right? With an amine group, NH2 group. Let's go with this. Replace with the amine group. Amine group. I'll replace this hydrogen with a functional group called as R group. Functional group called as R group. Only the hydrogen is left out. Only the hydrogen is left out. So what do you get? We end up getting a structure which resembles carbon, COH, NH2, hydrogen, as well as your R group. This is a typical structure of a amino acid. Typical structure of a amino acid. Okay. Now it's given us here. Amino acids are organic compounds containing amino group and acidic substituents on the same carbon. The alpha carbon hence is called as the alpha amino acid, the middle one. They are substituted methanes. You take a methane and you completely substitute it. There are four subunits. First one is what, tell me students? Hydrogen, carboxylic group, amino group and a variable group. Four different groups. That is, if you have a carbon here, COH, NH2, H, and R. This is a typical structure of a amino acid. Based on the nature of R group, there are many amino acids. For example, if the R group varies, now if I keep the R group as hydrogen, it becomes glycine. Now if I make this R group as your CH3 methyl group, it becomes alanine. Alanine. Or for example, if I take this R group and make it hydroxymethyl, CH2OH, hydroxymethyl, it becomes serine. Serine. So based on the R group, which R group is present, a particular amino acid can vary or change time to time. Okay. Now, I think it's written here. No? Now I'll teach you something called as essential and non-essential amino acid. What is the difference between essential and non-essential amino acids? What is the meaning of essential? What is the meaning of non-essential? Essential amino acids and non-essential. Essential amino acids are obviously are important, but not produced by the body. Not produced by the body. If they are essential and not produced in our body, how do we get such amino acids? Such amino acids can be taken with the help of diet, the food. That's why I tell, right, eat a lot of fish, eat a lot of protein, they say, right? This is the protein and amino acids. So are the amino important but not produced by the body. Essential. Non-essential, we require them, but they are produced by the body. They are produced by the body. So we don't have to take externally the non-essential amino acid. If essential amino acids are there, it's well and enough. Now you should know one more thing, students. How many of you know this? People who go to gym, they drink something called as protein powder. Now before what used to happen is there used to be a big scam. Big scam used to happen before. Okay. What is the meaning of scam here? There's always protein boxes, right? Protein dabbas. In that, instead of giving essential amino acids, they used to completely fill it with non-essential, and they're selling it to you until it was completely told later on that wait a minute, these amino acids are not even required for my body because they're already present in my body. I don't have to take it from outside. So later on, all the protein companies started telling the which amino acid they have added into the protein powder, and most of the amino acids should be of essential amino acids. Why? Because they're not produced by the body. Okay, they're not produced by the body. Beautiful. Any doubt here, students? Any doubt here regarding amino acids? If there is no doubt there, let's go to the next concept. What is amino acids can be broadly of two types, friends. Okay, amino acids can be broadly of two types. One is called as your aromatic amino acids. Other one is called as aliphatic. Other one is called as aliphatic. Aliphatic is also called as non-aromatic. Non-aromatic amino acids. Example, students, aromatic amino acids will have benzene ring. Benzene ring. For example, this is the aromatic amino acid. Will have benzene ring. While non-aliphatic amino acids will have straight chain. They'll have straight chain here. Okay, that is the major difference. See here, aromatic amino acid, phenylalanine, benzene ring is present. Tryptophan, benzene ring is present. Tyrosine, benzene ring is present. They are the aromatic amino acids. Now, students, amino acid can also be something called as acidic amino acid, basic amino acid, neutral amino acid. What is the difference? Understand. In your acidic amino acid, it will have one extra COH group. See here. If you look at the structure of aspartic acid, glutamic acid, see, first of all, a normal amino acid will have one COH group. Both aminos have one COH group, carboxyl group. So if an amino acid has more than one COH group, more than one carboxyl group, this is the second one. This is the second one. If it has more than one carboxyl group, it becomes acidic amino acid. More than one carboxyl group. Okay. What is basic amino acid? All of you know an amino acid has an amino group. Can you see here? This is the amino group, amino group, amino group. If a particular structure of amino acid has more than one amino group, as here in this case here, in this case here, and in this case here, if it has more than one amino group, then it becomes a basic amino acid. Then it becomes a basic amino acid. Okay. Your neutral amino acids will not have any extra carboxylic or amino group. That is neutral amino acid. So, Shwan, tell me how many amino acids are there totally? How many total amino acids are there? Total amino acids. You will tell me, sir, there are 20 different types of amino acids. Out of these 20 amino acids, 3 are acidic, 2 are basic, and 15 are neutral. 3 is acidic, 2, this is 2, sorry, 2 is acidic. Aspartic acid and glutamic acid. 2, 3. Basic is arginine, lysine, and histidine. 3 different types of basic amino acid. Tanuja, what I'm trying to tell you here is, if a particular amino acid has more than one amino group, see, one is always there. If it has more than one, it becomes basic. You know, so one is there, one more is here, so it's becoming basic. One is here, it has one more type of amino group, it becomes basic amino acid. Neutral amino acids will not have anything like that. Neutral amino acids will have something called as no extra carboxylic group, no extra amino group. In the case of neutral. In the case of neutral. Understood basic now? Basic will have one extra amino group. See, this is the general formula, right? This is the general formula. NH2, C and R. Now in this R part, if we have an extra amino group, NH2 group, extra NH2 group, then it becomes basic. If it has an extra COH, acidic. Understood now? Understood? Yes or no? Tell me. Yes or no? Basic will have an extra basic NH2. Acidic will have an extra COH. Neutral will never have anything. Neutral amino acids will not have a extra COH or extra NH2 group. Okay. Hmm. The R group or in this proteinaceous amino acids can be hydrogen, glycine. Methyl group, alanine. Hydroxymethyl, serine. Very important line. We learned it here. H, glycine. CH3, alanine. Hydroxymethyl, serine. Learn these three, they can ask you a question on this. Structure wise, they can ask you a question. Hmm. See here. The chemical and physical properties of amino acids are essentially of amino group and R is the functional group. Based on the number of amino and carboxylic group, number of amino and carboxylic group, there are acidic amino acids. They'll have more number of carboxylic group. Glutamic acid, aspartic acid, basic and neutral. Neutral, they have given example is 15. Valine they have given, you can take any 15 of neutral amino acids. Okay. Aromatic amino acids, tryptophan, phenylalanine, tyrosine, and you have aliphatic amino acids also. Now, tell me now. Sometimes what happens is when you're watching your favorite movie, you're very happy. When you're watching your mother's favorite, when the mother is watching serial, you're like, why this mother watching serial? I wish I could watch the TV, right? So what is happening there is you are happy when you are in a good surrounding or you're in a surrounding which you like. You're not happy when you're not in a surrounding which you don't like. It changes. So basically, all of you change with the surroundings. All of you completely change the surroundings. If the surrounding is good, you'll be happy. If surrounding is not good for you, you'll be very sad. That change in the surroundings, friend, that change in the surroundings based on the change in the mood based on surroundings, that you will understand with the help of that. The same concept will apply in the case of vitriol. For example, if you take a amino acid and keep it in acidic medium, it will behave very differently. If you take an amino acid and keep it in basic medium, it will behave very differently. Very differently. So when you keep the amino acids, when you keep the amino acid in a very special pH, it is mostly near a neutral pH. Okay. So when you take an amino acid, when you take an amino acid and you keep it in such a pH that this particular hydrogen is going here. Hydrogen is forming a bond with NH2. What will happen now? Tell me. This will become NH3 plus C hydrogen, CO minus and R group. So what we learn? Whenever in a very specific pH, what is happening? At a particular pH, we have intramolecular proton transfer. Intramolecular proton transfer. Transfer of proton within the amino acid. And this will happen at a particular pH. The pH is called as isoelectric pH. Isoelectric pH. And this structure right here, which has a positive end and a negative end, and net charge is 0. This right here is called as your amphoteric ion. It can be called as amphoteric ion or Zwitter ion. Zwitter ion. Okay. So that's how we are obtaining this Zwitter ion. Intramolecular proton transfer. Intramolecular proton transfer is happening to form the Zwitter ion. Okay. See. A molecule with one functional group having the positive charge, other one having a negative charge. Net charge is 0 at isoelectric pH. Okay. That is the end of proteins. That is the end of proteins. Any doubt here? Tell me. What did we learn till now? We learned about Zwitter. This is Zwitter. You see here. Negative, positive. We learned about aromatic amino acid. We learned about the structure of amino acid. Acidic, basic and neutral amino acids. Aromatic, aliphatic, structure, essential, non-essential, and the three different structures. And the three different structures of different amino acids. So with that, the first part of the chapter is over. I hope there was no doubt till now. Every element was clear to all of you, and similarly, we will do the second part now, and that is the end of today's class. After lipids, I will stop today. Okay, don't worry. I will not take a lot of your time. We will do short videos to make sure you understand a lot. Okay. Now we will do something called as lipids. Understand lipids. What exactly is lipid? What exactly is lipid, friends? Lipid. What exactly is a lipid? Lipid is nothing but an organic compound is an organic compound whose molecular weight is less than 1000 daltons, around something like 800 daltons. Even though it has less molecular weight, it is placed where? It is placed in the retentate on the top. Acid insoluble fraction on the top, it is printed on the top. Okay. Now here you understand here is how do they do it? They do it with a simple mechanism. Each one is a lipid. Each one is a lipid. All the lipids are joining hands. And you see, all the lipids are joining hands and they are making one entire gang. When they make an entire gang like that, it becomes very difficult for them to pass through the muslin cloth. So that is why students, that is why your lipids, even though they are acid, they're supposed to be molecular weight is supposed to be less than 1000. They're supposed to come to filtrate, but they will never come to filtrate. They'll be present on the top only. Okay. Now, students, understand something called as the two different types of lipids. The first one is called as fatty acid. Fatty acid. The other one is called as glycerol. Fatty acid and glycerol. In fatty, what is fatty acid? Fatty acid is nothing but carboxylic acid plus an R group. Plus an R group. This R group can be anything. For example, C double bond O H is here. To this carbon, we also have an R group also attachment. This R group can be anything. When I say anything, I mean this R group can be basically a methyl, or it can be ethyl, or it can be from C carbon number 1 to 19. Any carbon it can be. Any carbon it can be. That functional group is here. Okay. Based on the functional group, the name of the fatty acid can vary. 3 fatty acid, tri. 4 quadra. 5 penta. Right? This is your carboxylic acid. This is your fatty acids. Fatty acids. Okay. Now, friends, all of you should remember this. Fatty acids can be of two types. Fatty acids can be of two types. Okay. Fatty acids can be of two types. Saturated and unsaturated fatty acid. What is saturated? Saturated fatty acids will have single bond. Unsaturated fatty acids will have what? They'll have something called as double and triple bond. Double and triple bond. Yes, saturated fatty acids, their melting point is relatively higher. Here melting point is little lower. In the case of saturated fatty acids, they're solid at room temperature. For example, butter or ghee. Butter or ghee is more of solid at room temperature, while your unsaturated fatty acids are liquid at room temperature. Basically, gingerly oil, the cooking oil, the cooking oil. Basically, the cooking oil. Okay. Basically, the cooking oil or gingerly oil. Okay. That is basically your fatty acids. Now, what about glycerol? What about glycerol? How is glycerol different? And what do you see in glycerol, students? What do you see in glycerol? Now, glycerol is basically an alcohol. It is basically an alcohol. Okay, it is an alcohol. It is an alcohol OH group. OH group. To be precise, it has three carbon, 5 hydrogen, and 3 OH. That is two, 3 carbon, 3 OH, and 5 hydrogen. One here, one here. 1, 2, 3, 4, 5. 5 hydrogen. The other name for your glycerol is called as trihydroxypropane. Trihydroxypropane. Okay, trihydroxypropane. This is your glycerol. This is your glycerol. Okay, this is your glycerol. For example, students, glycerol has special function. For example, don't forget esterification. Glycerol, if I take this a glycerol, let's take a glycerol here. 3 carbon, OH, OH, H, 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 and H, and H. Now what will happen what will happen if there is, if I remove one of the, if I remove one of the OH and if I take one of the OH out and you bind it with you bind it with a fatty acid. If you bind it with a fatty acid, what will happen? One of the OH, you take it to fatty acid, you add a fatty acid here. See what want to? If you add fatty acid to one of the OH, that becomes a monoglyceride. You add here also one more fatty acid, you add diglyceride. One more fatty acid here, that becomes a triglyceride. So you're taking a fatty acid, you're taking a glycerol, and you're joining them together. Okay, you're joining them together. If you take one part monoglyceride, diglyceride, triglyceride. Right now, this is a monoglyceride. Okay, monoglyceride. They're called as glycerides. Glycerides. What are glycerides? Glycerides are nothing but your Glycerides are nothing but your fatty acid plus glycerol joining together. Fatty acids and glycerols joining together. That is a glycerol. Okay. See here. Another simple lipid is called as glycerol. Glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids together. Okay. They are found esterified with glycerol. When they are present together, they are called as esterification. They can be monoglyceride, diglyceride, or triglyceride. They're also called as fats and oils based on the melting point. Oils have lower melting point, gingerly oil, okay, and hence remain in oil in winter. Very important lines here. Phosphorus and phosphorylated organic compounds in them. That is, some lipids will have phosphate attachment also. A lipid will have phosphate attached to it. Such a phosphate attached called as, they are called as phospholipid. So phosphate plus lipid, phospholipid. Example is lecithin. You see here, lecithin is a simple example. Lecithin is an example of phospholipid. Okay. Some tissues, especially neutral tissue, have lipid with more complex structure also. Example is your cholesterol derived lipid. Okay. Students, that is the basically the different types of lipids. Now I think we will do even the nucleic acid. Very easy to understand nucleic acids. What is nucleic acid? Nucleic acids are basically the living tissues, right? Now, living organisms have number of carbon atom in the heterocyclic rings and can be found in some nitrogenous bases. Friends, this is a nucleotide. A nucleotide will have a pentose sugar here, a phosphate and nitrogenous base. This entire thing is called as nucleotide. Then what is a nucleoside then? Nucleoside is what, nothing but if it has only sugar and only base, that is a nucleoside. If, what is a nucleoside, students? A nucleoside will have what? Nitrogenous base plus sugar. Both, if I add even phosphate to it, if I add phosphate to it, it becomes nucleotide. Nucleotide. So nucleotide will have all three of them. Nucleoside will have only both of them. Both of them nucleoside. Okay. Simple difference everyone should know. Now, we have something called as nitrogenous bases. There are mainly 5 types of nitrogenous bases. 2 are purines. Purines. That is, guanine and adenines are purine, which are double ring structure. Can you see here? One ring, second ring. One ring, second ring. 2 ring structures. That is the question, famous question. The double ring structures are present where? Purines. Okay. At the same time, if you do pyrimidines, what is the trick here? Trick is very simple. Cut the pyramid. Cytosine, uracil, thymine. Cytosine, uracil, thymine. They are single ring structures. One ring structure. Can you see everywhere one ring? Pyrimidines. Purines and pyrimidines are nitrogenous bases. Okay. See here, carbohydrates, we learned carbohydrates. We didn't learn, sorry, amino acids. Glycine, alanine, serine, all three are present here. Glycine will have hydrogen, alanine will have CH3, serine will have CH2OH. Palmitic acid, aliphatic it is straight. Palmitic acid, fatty acid it is. Glycerol, trihydroxypropane, 3-hydroxyl. Glyceraldehyde. Can you see? Every single OH is replaced by a fatty acid. One more OH also replaced by fatty acid. One more OH also replaced by fatty acid. This R here. You see R, R, and R. Fatty acid. Triglyceride. We have phospholipid, which is an example is lecithin. Phospholipid, which is present in the cell, which is present in the plasma membrane. Present in the plasma membrane. Cholesterol is a derived lipid. Derived lipid. Nitrogenous bases, pyrimidines and purines. Nucleosides, adenosine, guanosine, and unosine. Nucleotide will have what? Ilic acid. Adenolic acid, uralic acid. That is nucleotide. Nucleotide. Do we miss anything here? Huh. We found attached to the sugar. They call as nucleoside. Nucleoside will have only what? Nitrogenous base and pentose sugar. If phosphate group is also found esterified to the sugar, then it is called as nucleotide. Okay. So we have adenine. So we have DNA and RNA are the genetic material. Basic, all of you know. Here you should also notice how do you differentiate between nucleoside and nucleotide. Can you see here? DINE, SIDE, adenic acid. See, adenic are nucleotides. Adenosine, guanosine, thymidine, uridine, they are nucleosides. They are nucleosides. Ilic acid, thymidic acid, guanic acid, uric acid. That is a nucleotide. A nucleotide will have acetic acid attached to it. Okay, acid attached to it. Clear? Beautiful. There is a simple difference. Now, students, let's come to last part of today's class. That is nothing but your what is the meaning of primary and secondary metabolites? Very simple to understand, students. Very, very simple to understand. In the case of plants, there are some metabolites which are needed by the plants. They require it. Without those pigments, a plants cannot survive. Okay. Hello, Mahima, Radhe Radhe. And you cannot survive. But there are some metabolites in the case of your plants which we can survive without. And the secondary metabolites are produced in the case of plants as a defense mechanism. As a defense mechanism. For example, you see here, list of biomolecules. See here. If we one were to make the list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugars, everything. They are called as metabolites. In animal tissues, one noticed the presence of all such categories of compounds shown are called as primary metabolites because they are required by the plant for their development and growth. However, one analyzes plant, fungal and microbial, one would see thousands of compounds other than the primary metabolite are also present. These are particular structures which are present other than the primary metabolite is called as secondary metabolite. Example is what? Alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scented gums and spices. All of these are secondary metabolites if you can see here. Primary metabolite have identifiable function and play a role in the normal physiological process. Primary metabolites can perform simple function. For example, carbohydrates can give energy. Lipids can form some function. Protein can form some function. It's very fixed. It's very fixed. But when you come to secondary function, secondary metabolites, the function is not known. Function is not known. Here is a table, friends. Understand this table. By heart this table at any cost. Pigments, carotenoids, anthocyanin. Alkaloids, morphine and codeine. Codeine is used in your cough medicine and all. Morphine is used as a, to relieve the pain and all in cancer treatment. Terpenoids, essential oils. Nice smell will come. No, essential oils, lemongrass and everything. Toxins like arbutin and ricin are toxins. Lectin, concaveolin A, which is also a plant growth regulator. Not plant growth regulator, it is a cell developer. It's a mutagen which promotes cell division. Promotes cell division. Drugs like vinblastine and curcumin. Curcumin from curcumin longa, vinblastine from vinca rosa. Vinblastine also is a cancer treatment drug. Okay, gums, rubber, and cellulose. All of these are called as secondary metabolites. Each one of them role is not known, but each one of them has an amazing benefit which we use as humans. Okay, so a lot of questions have come from secondary metabolites. So learn the secondary metabolites, students. So I think we will. I think this is we. I think I taught you all this right already. Or we can read this. Yes, yes. We know what is biomacromolecule. Present on the retentate acid insoluble fraction more than 1000 Daltons size present on the top. That is what here. Can you see here? Biomacromolecules. Okay. Acid insoluble fraction. Only four types are there. What is that? Proteins, very heavy. Nucleic acids, very heavy. Polysaccharides and lipids are also very heavy. Lipids might not be very heavy, but since they are forming a gang, they're present on the top. They're present on the top. These classes of compounds, with exception of lipid, see, exception of lipid, have molecular weight in the range of tens of thousands of Daltons. Very, very high weight. Okay. Now, of this, which molecular weight is less than 1000 are usually referred to as micromolecules. Less than 1000 is micromolecules, more than 1000 is macromolecules. Okay. While those are found in the acid insoluble fraction on the top, they are called as biomacromolecules. The molecules in the insoluble fraction, with exception of lipid, all of you, 800 Daltons. Why? Because they form vesicles, right? Polymer substance. So lipids are not indeed small molecular weight compounds present not only such as the but not only as such but also arranged in structurals like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. The cell structure and other membranes are broken into pieces and form vesicles, which are not soluble, which are not water soluble. So vesicles are nothing but lipids. All the lipids are joining together, hence, and they're present as a group, and the group is not coming down to the filtrate. That is the end of the story, students. Okay, so this line here, can you see? Lipids are not strictly macromolecules because the size is less than 1000 Daltons. Size is less than 1000 Daltons, but since they are forming vesicles, they can be found in macromolecules on top. Okay, they're found as macromolecules on top. So acid soluble pool represents roughly cytoplasmic composition. Macromolecules from the cytoplasm organelles become acid insoluble fraction. Everything on the inside cytoplasm, acid insoluble. Okay, it's given here. Average cell composition: water, proteins, carbohydrates, lipids, nucleic acids, ions. So with this, you get an idea about, from today's chapter, you get an idea about what is a micromolecule, what is a macromolecule, what is an acid insoluble, what is acid soluble, what is a filtrate, what is a retentate. You also got an idea about what is a amino acid. All the aminos join together, they make up the protein. The structure of amino acid, how they can vary based on the functional group. Acidic basic amino acid. What is the aromatic amino acid and what is the zwitterion? Okay, all of you know that. You also learned something called as lipids. You also learned something called as lipids, fatty acids and glycerol. And if they join together, they make up glyceride. They make up the glyceride. You also learned something called as nucleotide and nucleoside. You learned what is a purine pyrimidine. So all this was learned in today's class, students. To summarize the entire thing which you learned today. Now in the next class, we will do the remaining part of it. And until then, students, take care all of you. Smash the like button and I will see you in the next class. Bye bye all of you.